Welcome now to Dr. Jeffrey Shaman, Director of the Climate and Health Program and Professor from the Mailman School of Public Health at Columbia University. Dr. Shaman, it's so great to have you with us today, and we look forward to speaking with you more on your study.
Thank you for having me, Mabel. It's a pleasure.
Now, you've been, as I said, studying these coronaviruses, other ones for the past 4 years. We don't hear a lot about them because they cause common colds, and people are paying a lot of attention to that. But now that you have that information, how does that inform you on how this pandemic will pan out?
Well, what we're looking for, because this is a novel virus for which, it's like an alien being is dropped down in our midst, and we have to figure out what it is, what it does, what makes it tick, how it operates, and how to control it. We also want to look to see if there's anything that we've had prior experience with that gives us any insight into how this one may behave. So humans have been infected by 6 other coronaviruses of which we are aware. There may have been others in the past, but we haven't documented those. 4 of them are these endemic coronaviruses. They go by these funny alphanumeric names like OC43 and HKU1, and they tend to cause common colds. But they have a number of characteristics that we can look at to try to understand if they have any bearing on what's happening and what might happen with SARS-CoV-2, the causative agent for COVID-19. Among them, there is the fact that they're seasonal. We see that in temperate parts of the world, such as in the United States, they occur in the wintertime. They tend to peak in January or February, and then they disappear, or they're very low levels, in the summertime. And it's a seasonality that's very similar to what we see with influenza. So influenza is very similar, both seasonal influenzas and even the pandemic strains that have come through and emerged, we've seen they have this seasonal component to them where they tend to peak in the wintertime, and then they tend to disappear or be less transmissible during the summertime. Mm-hmm. Now, the reality is that we've studied flu a lot more than we've studied these endemic coronaviruses, these 4 viruses that have been circulating that cause typically very mild colds in most people. So we don't have a lot of studies as to why those endemic coronaviruses are seasonal. What we do know, because they mirror what we see in flu, is what goes on with flu. We've studied that in greater detail, all the way back to the 1940s, in fact. And what we see there is there appears to be a sensitivity of the influenza virus to humidity conditions. You might ask yourself, well, why does a virus care about humidity conditions? Well, the way respiratory viruses are transmitted is that when an infectious person sheds the virus, and you shed the virus in your oral and nasal mucosa, you expel it out into the environment when you're breathing, when you're speaking, when you cough, when you sneeze, in these respiratory virus droplets. These droplets that contain respiratory virus, I mean to say. Mm-hmm. And some of those droplets have to survive long enough and find their way to another host in order to set up a new infection. While they're actually out in the environment, once they're expelled from your internal system and then your lungs into the environment, they are subject to the whims of the environment. Some viruses are very sensitive to heat, some to cold, some to sunlight, some to chemicals. The way a respiratory virus is transmitted is a little bit of a puzzle. So we don't actually know how respiratory viruses are transmitted, which is very weird. We know how lots of other diseases are transmitted. We know how sexually transmitted diseases are transmitted. We know how mosquito-borne diseases are transmitted, and we know how diarrheal disease is transmitted. Respiratory viruses, we know how they can be, but we don't know what the dominant mode is. And this is important, and it's important in the context of COVID-19. So if I'm sitting here and I'm speaking to you, unfortunately, you're not in the room with me, Mabel, but I'm spewing out droplets all the time. We all are. The droplets I spew out when I'm speaking tend to be smaller. If I'm contagious, some of them will have viruses in them. And they will be small enough, perhaps, that they'll remain aerosolized on the turbulence in the air, and they'll float around, and somebody else can come along and inhale it, and that would be an airborne mode of transmission. Or if I was, in fact, person to person with you, Mabel, unfortunately, some of that spray would hit you, and some of it might get in your eyes or your nose or your mouth, and that might transfer the virus to you. Lastly, some of it might settle on the table that's between us when we're talking. And you might then touch the table, and then you might touch your face, your eyes, your nose, your mouth. You might eat a cheeseburger, and that might transfer the virus to you. While the virus is out there, it's exposed. and it can degrade. Viruses are really weird organisms when you get down to them. They're these strands of nucleic acid surrounded by protein capsule, lipid layers, and such. There's not much to them. They're not really alive, many argue, and I would agree with that. But they are subject to being broken down, to breaking apart. The nucleic acids getting degraded by RNase, or the proteins or the lipid layer breaking down, something happening so that it doesn't survive and it's no longer viable and capable of entering a new host cell and setting up a new infection. So while it's there and while it's moving in the medium between hosts, it's subject to some of those environmental conditions. And it may be that humidity conditions modulate the survival and viability of some respiratory viruses. Certainly for influenza, there seems to be evidence that that is the case. And we get this evidence out of laboratory experiments, as well as statistical examinations of rates of infection and whatnot. The laboratory experiments seem to be the most definitive because they're very controlled, even though they're somewhat idealized. And what we can see there is that the virus remains viable longer when the humidity in the air is very dry. And we don't know why. We don't know the physicochemical reason why, but it seems to affect both the viability of the virus, its ability to transmit amongst animal models, and we see it certainly because we get the flu in the wintertime, and in the wintertime, the air is much drier, both indoors and outdoors, relative to summertime. So it would seem that maybe that's responsible for what's called this phase locking, where we get the flu in the wintertime, and we don't see as much of it in the summertime. Mm-hmm. Now, for the coronaviruses, these endemic coronaviruses, we see that they have the same cycle, but nobody's done these experiments to see, is this related to temperature or humidity or sunshine or people being indoors more often in the wintertime, or any of a number of other hypotheses that are out there. So we don't know what drives those coronaviruses. And for this new virus, nobody's done the experiments really as of yet. There's been a limited number. There's been some indication. As a matter of fact, President Trump at a White House briefing about 4 or 5 weeks ago had a table that supposedly showed analyses done in a DoD lab. I haven't seen those published elsewhere, but they're very consistent with humidity being the main driver, other than sunshine, of whether or not the virus survives.
Okay, now we're heading into the Memorial Day weekend here, and some temperatures in some areas of the Northeast, for instance, will reach in the upper 80s. How carefully will you be assessing those conditions when studying this virus?
We have to keep looking at it over time. We have to, what I'd love to see are some more laboratory experiments. In the absence of those, we have to keep monitoring what's going to happen. There are some cautions as to whether or not this will be seasonal. So when we had the 2009 pandemic of flu, there was a spring outbreak in the Northeast, and then it stopped in late May, really in mid-June. It was done. And in July and August, there was very little activity in the United States. And then as things started to cool off and the humidity started to drop, it came roaring back, and we had this massive wave in the fall.
Okay.
That's happened with other pandemics as well. And that's a concern here for COVID, that we may see something like that with this virus. But if you look at places around the world, you see that there's activity, there's a lot of transmission in some very hot and humid places. Like Manaus, Brazil, the capital of Brazil, is having a huge outbreak right now. It's 3 degrees off the equator. That whole area around it is seeing a lot of it. Singapore struggled with it for some time now. It's not clear that whatever modulation by humidity conditions is afforded that reduces the transmissibility of the virus is enough to stop it. This virus is actually innately more transmissible than influenza.
Is it misleading to presume that people who have had COVID-19 and then recovered have the antibodies that will protect them, based on what you know?
We'd love to think it. We'd love to think that we're basically one and done with this virus, so that you're infected with the virus, you develop antibodies, memory T cells, other components of your memory system, and the next time you encounter the virus, it takes it and it removes it from your body and preemptively clears it so you're not infected. The reality is we don't know. We still don't know as of yet. Unfortunately, for these other coronaviruses, the endemic ones that we get the common cold, we see that people have a lot of antibodies. 90% of the population has antibodies for each of those 4 coronaviruses. Okay? But we get them repeatedly. And we have evidence to show that people get repeat infections by these coronaviruses, which is very concerning because it means they're getting them over and over again in spite of some antibodies that are developed. Now, we don't know, once again, a lot we don't know here, whether that applies to this new coronavirus. So the three key questions are, can people get infected with this virus more than once? If they can, how often? Are you going to get it every year? Are you going to get it every 5 years, every 10 years? Those are really important to know. And also, if you are subject to repeat infection, are your symptoms going to be less severe typically? Or are they going to be more severe? We really need to answer those questions. And unfortunately, only time is really going to tell, because we're going to be able to see whether or not there indeed are repeat infections definitively.
Well, we did see already reports, Dr. Shaman, about sailors aboard an aircraft carrier testing positive for a second time.
Yep.
Are you perhaps suggesting also that maybe the testing is a little bit quirky, that it's not definitive that this is coming back for a second time? Because
it is not definitive. So yeah, that's exactly right. So it may be that the testing was a false positive, excuse me, was a false negative. I got it right the first time. And that in fact, those people maybe just had been in a low period of shedding and they'd not cleared the virus from their body, and it's the same infection. Or it could be that indeed it was very, very low and not detectable. It was below the limits of detection of the test. And then the virus is somehow relapsing within their body, and it's actually producing more copies again. We don't know if it's one of those possibilities, some kind of recrudescence, or if it is, in fact, a repeat infection. It's not definitively shown. This also happened in Korea and China. So we've seen this over and over again, where people who we really thought were cleared and had tested negative are testing positive subsequently. But we haven't found really definitive evidence that it is a repeat infection for these individuals.
Now, certainly many people are concerned that we don't have a national testing program in place, or even contact tracing nationally. So will we have squandered almost 10 weeks of lockdown since we don't have that in place and states are starting to reopen?
And that is another critical question. If I had a crystal ball, I could tell you. We have a very challenging problem in front of us. The hard problem is this: we want to suppress the virus and keep it at very low levels as best we can using non-pharmaceutical interventions, mask wearing, social distancing, school closures, limiting of gatherings, etc. And simultaneously, we want to disrupt our economy as little as possible. We want to try to keep it going so people have employment, they have work, they can earn money, they can feed themselves, and that we don't suffer the long-term consequences of that kind of disruption that takes place. That is a really hard problem to find the solution to. When you look around the world, you see that a lot of countries haven't yet met that challenge. They've not been able to navigate that. There are some that have, however, and they are the countries that I think we should be looking to try to model our patterns of control and resumption of activity after. And they include places like South Korea and Taiwan and New Zealand and Iceland and Vietnam and Thailand and Germany and Israel. If you look at what these countries have done, and they've had to deal with large outbreaks in some instances, particularly Germany, South Korea had a very large one early on. They've really managed to navigate this a lot better than what we've done in the United States. Mm-hmm. So there is this concern that you don't want to just go back to economy and just say, oh, forget about it. Let's just go back to a full economy and we'll suffer the consequences. There is a developed country that is actively doing that. That is Sweden. They have chosen not to disrupt their economy and let the virus move through them. They're only recommending that their citizens stay home if they can. They've shut off some access to long-term care facilities, and they've shut some schools. But for the most part, they are doing their normal activities. And they have a very high number of deaths given their population at this point.
Okay.
We would like to see something, I think, more like what's happened in South Korea. They kept their economy open while they did this. They had face mask use. They had a real advantage in South Korea. Make no bones about it. They already had legislation in place that allowed them to contact trace and go into people's credit card records and their cell phones to find out who they've been in contact with if they tested positive for a pathogen. They put that on the books 5 years ago after seeing what happened with Ebola And it was even more reinforced when MERS, which is the Middle Eastern Respiratory Syndrome, another coronavirus, emerged in their country for a time. They also have a quarantine act, which allows them to legally keep people home if they've been ordered to be home because of quarantine. So they close their schools, they keep the businesses open, face masks are used, and they manage to really crush down this outbreak. They take it down to very low levels. They had a big outbreak and they bring it down to levels where they're seeing single digits numbers of cases at that point.
But isn't Korea facing another outbreak now?
Great question. So they went from single digits now to about 20 to 30 cases a day about 10 days ago because they started reopening their economy further. And I'm not going to try to minimize it and say that there wasn't damage to the economy because of the outbreak. People weren't going to restaurants the same way. There were all sorts of problems associated with it that actually made them take a hit. But they reopened baseball season. They've opened their churches up. They opened nightclubs and bars back up. And it was at these nightclubs and bars that they seem to have had a cluster outbreak. And now they're getting 25 cases a day.
Okay.
Now I want you to contrast that with what we have in the United States. Their resurgence of activity that you see there, if they were to have 25 cases a day for the next 1,000 days, which is almost 3 years, that would be 25,000 new infections. We are getting that every day in the United States. Korea is a country of 51 million people. We're only 7 times larger than that in terms of population. So they could stand for 1,000 days and 0.05% of their population would be a confirmed case. If you were to say maybe 10 times we're infected, that's 250,000 people. That means over 3 years, 0.5% of their population is infected while they're maintaining their economy and they're buying time so that an effective vaccine or therapeutic can be developed and deployed, which would allow that 99.5% of the population that hasn't been infected never to run the risk of having the disease.
And doesn't your math also say that if we had started some of these more stringent policies just a week earlier, the numbers would look a lot different today?
Yes, it does say that. They would look a lot different today if we were to do that. The reality is earlier that you grab onto this virus, that you aggressively try to control it and do things to stop it, the better off you'll be. And the important thing,
may I ask, would that also include banning mass gatherings after lockdown is lifted? Super spreading events.
I think mass gatherings are particularly a concern. I think particularly indoor mass gatherings are problematic. So there are problems associated with bars, problems associated with concerts, problems associated with having sporting events with an audience, problems associated even with churches that really need to be thought out well, and places of worship.
Mm-hmm.
The reality is those seem to be events where you bring enough people in. If somebody isn't efficiently contagious and spreading the virus, they have the opportunity to give it to a lot of people all at once.
What about going to the beach? What about being outside, going to the beach during the summer months?
I actually think that the beach is not a high risk personally. Now, I could be wrong about this, of course, but I think that the virus, given the indication we saw from that Donald Trump's table at the press conference, it's not inconsistent that it would be pretty wimpy about being exposed to sunlight. And being on the beach on a sunny day where families are spaced out, there's enough room that they're not all crowded and packed together, 6 feet apart there, generally further apart, I think it's probably not the most dangerous environment. I think we have to understand, though, regardless, anytime you're getting together with people and mixing with people, you're running some level of risk. It's a question of what is a low probability transmission gathering or event, and what is a higher probability. We want to minimize the ones that we think are the more dangerous for supporting the transmission of this virus.
How could your research impact the development of an effective vaccine?
My research is more on the epidemiological side. I'm not in vaccine design or therapeutics design. One way in which the type of work that I do, which involves a lot of modeling and analysis and some epidemiological field studies, can help is to try to figure out how to design the clinical trials or where to implement the clinical trials and when. In other words, trying to find a way to make it happen as expeditiously as possible simply by holding the clinical trials in areas where you think it'll have maximal effect. Where unfortunately, where you think there may be a lot of activity, where you're going to be able to see that some people didn't get the virus and others did when you look across some randomized clinical test design.
And how should people think about learning to live with this virus? What does that mean? And what does the infrastructure look like for that? And do we have any of that in place now?
Yeah, that's a great question. I think, look, we're all holding out hope that we'll have as soon as possible a really effective vaccine or an incredibly effective therapeutic. That's sort of the game changer, the get out of jail free card that would allow us to deploy it and make it so that a huge majority of the population is no longer going to be at risk of death because of acquiring this infection, right? In the absence of that, until we get that, we need to try to find a way to navigate that hard problem I talked about, suppressing the virus while simultaneously minimizing economic disruption. And what that means is some kind of new normal, right? It clearly, this type of virus has enormously disrupted some industries, the airline industry, the hotel industry, vacation industries. They are really being decimated by this. And it's a huge problem for them. And how we're going to reboot that is going to be a very challenging problem for economists and government leaders to figure out as we're coming out of this. It doesn't appear that it's time to do that. At the same time, sheltering in place for another 6 months or a year while we wait for a vaccine seems like a very, very hard ask. We need to find a level of constraint that allows people to have a reasonable amount of activity so that they can support themselves economically, psychologically, emotionally. We need to have this because that's what we are. We can't just be living in our caves like hermits waiting for this to end. That's a huge ask that we prefer not to go through. Now, if this were an extreme virus, maybe that would be the ask. But the reality is we are seeing a handful of societies around the world, that have managed to actually navigate this problem. When you look at what, I'm going to focus again on South Korea, what Germany seems to be accomplishing, when you look at those countries, and I pick those two in particular because they're large, 50 million, 90 million people in them. These are big countries and they're landlocked. They're not island nations. They don't have this ability to be surrounded by water and really insulate themselves as much. And they're able to actually maintain a semblance of the economy, minimize the deaths that they've seen there. We have to start to model our control of this virus after that. We have to take on the testing practices and contact tracing that they've utilized, which is much more aggressive than what we've done in the United States today.
Sorry, go ahead.
Well, I was just going to point out that, if you look at the number of tests per infection identified, not per capita, but per infection identified, in South Korea, it's about 70 to 1. In the United States, it's about 8 to 1.
Wow.
Haven't tested as aggressively. We haven't looked as far. Now, it's gotten a lot further out of control here, and we have many more infections, and we've conducted more tests than any other country. But in terms of using it so we can not only just see the people who are coming to treatment because they're symptomatic and they need care, but to find people who might be infected, who suspect it by contact and don't have any symptoms, that's when it becomes very powerful. That's when you're really able to throttle this disease and limit its presence in the broader community. That's what active control is. Now, have to understand that contact tracing, even though it's boots on the ground epidemiology, is very, very powerful. This virus, when it infects somebody, it has a latent period, 3 to 5 day period on average, which the person is infected, but they're not yet contagious. Then that person will enter a pre-symptomatic phase where they're shedding the virus and they are contagious, but they have no symptoms for 2 to 3 days. That allows them to spread it without even knowing that they're infected, which is a dangerous aspect. One of the key things that allows this virus to get around. Then their symptoms start to develop and they start to grow. And eventually, after some days, maybe 2, maybe 7 days, they feel sick enough that they go to see a doctor and they might get diagnosed. So at that point that they're seeing a doctor, if they're testing and their positive test result triggers a bunch of people to go and say, who did you have contact with the last 2 weeks? And then to reach out to those people and ask them to quarantine themselves. Some of them, particularly within the last 4 to 10 days, may have been exposed to this person when they were infected, okay? And some of those people may still be in that latent period if they are infected. And by quarantining them, you've taken them out of circulation before they even start to shed the virus. So you really begin to squeeze it out of the population if you can gin up enough of this contact tracing in your localities to squeeze out the virus. It can be very, very powerful.
Without those powerful tools, what are your models telling you ultimately how many Americans will be infected with this virus?
You know, it's really hard to say because it's hard to know how much we're going to do to control it or when a vaccine or treatment will come. The worst case scenario is if we were to let this virus just do its bidding and just go through the population, we would expect around 200 million Americans would be infected. And we look at this virus and the estimates now are that 1 in every 200 or 1 in every 100 persons who gets this virus dies from it. So that means 1 to 2 million people will die from it in the United States, depending on where those estimates are and how correct they are. Again, assuming 200 million people get the infection. What we want to do is we want to keep a lid on it. Instead of having 20,000, 30,000 cases per day, it would be better if we had 200, obviously. Really crush it down, keep it really low. Because again, just like 1,000 days in Korea that I talked about, 1,000 days in the United States with 200 people getting infected per day is 200,000 infections, right? We could do that for 3 years and the majority of Americans would not be infected.
All right. Dr. Jeffrey Shaman, Director of the Climate and Health Program and Professor from the Mailman School of Public Health at Columbia University. Thank you. Thank you so much.